briefly, we have done a lot of work on these chemicals and how these chemicals affects the not only the ecosystem and even the other sources. And some of these chemicals we have taken for our study, and this analysis started from the microorganisms to man, where we have taken all the experimental models. Among them, we have also taken the rats, even the lower organisms, the microorganisms. Now, the evaluation of these pesticides toxicity, what we have selected. And these chemicals or the pesticides, if you analyze, there are nearly 3 types of pesticides: the carbamides, the organophosphates, and the chlorinated pesticides. Among these, even natural products also we use to control the different pests in the agriculture sector. So among all these chemicals, where the chlorinated pesticides are much more dangerous, and some of the experiments also we have done in the animals model, and where the chlorinated pesticides, the endosulfan plays very worst part in the country. And in Karnataka, so where it shows so many adverse effects, not only in the development, in other aspects. Now, I will not go in detail about all these things. So where you can see the organophosphates, the carbamides, and the organochlorines, and these are the chemicals. Among them, the organochlorines are very, very dangerous, including DDT, where we use to control the mosquitoes. And now they also got the resistance because continuous use of these things, where they get the resistance power and they are not afraid to DDT or any chlorinated pesticides. Exactly same thing what we also use the antibiotic resistance in most of the microorganisms. A wonderful chapter last year only we have published from USA, how these microorganisms get the resistance, that you take all the antibiotics started from penicillin, methicillin or vancomycin, where they are not afraid to any antibiotics. Even there are thousands of antibiotics what we discovered, and now these organisms get the resistance. So now one of my PhD students, who is a professor in biotechnology, now he also during accident where completely the whole skin has been scraped off, and till today, so those microorganisms got the resistance. Now the wound healing process is not taken place. So he got the PhD, and where lot of things he has worked on pesticides, and some of the work I am going to present in front of you, how these pesticides are affecting the humankind. Now this is the one which we have taken, the methomyl is one of the first the pesticides where you can use in the agricultural sector. So by using this one, where number of the side effects that you can see on this compound, we have worked a bit of this methophil and how this can also be degraded in the nature by the microorganisms. Now this is how the pesticides that has been spread to control the pests in agricultural crops, and you can see the spraying all those things when the farmer, not only the farmer gets inhalated by these chemicals, even some of the persons where they working in industry, they also affected by these chemicals. So once they inhale, even in women also what we have seen, so where the menstrual cycle also will be affected, it leads to oligomenorrhea or amenorrhea. So even the full complete sterility what we have observed, where you can see the sterile couples if both are working in the industries. Even in the male also it affects not only other physiological conditions, it also affects the fertility, and where it also causes sterility. Now this is another one, so where you can see in the agriculture how it is spread and how the man gets the inhalation. See, this is some of the report what we have seen from Karnataka, that Shimoga districts, where the chemical which is used tremendously, that is the glyphosate, where you can see the defects that you can see in the genomic level. So you can see the defects, but you can see the embryo and the tumor also you can see it induces a cancer. So where the chlorinated pesticides are the known chemicals where they induces the transfer the normal cell into a carcinogenic cells. Now this is another, you see, the victim of the endosulfan. So where also that affect you can see the bone, how the osteoporosis that has been taken place and the defects you can see, and also it affects the hormones, and also the neuro behavior also it changes. So that is the worst part what we can see. Now this is another report, and you can see the victims of endosulfan, Kasaragod, as just now I told, from Karnataka, and you see the defects that you can see make out. This is how the pesticide cycle that you can see in the environment. When you spray, so how it enters in the ecosystem, not only in the water reservoirs or not only in the rivers and oceans where it enters, and where you can see in the food chain, and ultimately the man has to think that if he discovered these chemicals, and ultimately it comes back to him. So this is the one, whenever we discover something and the man, before releasing into the environment or before releasing into the society, you must think 1,000 times how the effect it will be there. That one has to take care. So this is the cycle how that takes place in the nature, even in the aquatic, that is the ecosystem, where it enters through the cycle and enters the fish, and fish when the man eats, so these chemicals where you see the biomagnification and have the adverse effects. Now this is how the main part of any chemicals or the pesticides, if you take, where it acts on the cholinesterase or the neuromuscular junction, where this is the very important neurotransmitter, where this is affected by the chemical, and naturally it will affect the whole system, because these are the chemicals we use on the insects. So the insects are very stronger than man. So on one side the insects are evolved, and another side the man in pesticides has been evolved. So we have discovered thousands and thousands of pesticides. At the same time, now the insects also have got the resistance power, and they evolved wonderfully in the nature. Now they are not operating to any chemicals which you spray, because they got the resistance power. Now this is the mechanism. So the main objective you can see how it will be affected. So these are the things where on reproduction, on vital organs, biochemical contents, target enzyme interactions, oxidative stress, even steroidogenesis and behavior, and histopathological studies also we have done. Lot of work we have done how it will be affected. Just I am presenting you a few slides, so how you can see the dangerous of these chemicals. Now the investigation was divided to 5. The methomyl induced toxicity in testes, accessory organs, and methophil induced interruption of the vaginal toxicity, that means can be comparable to the menstrual cycle. So you will get this cycle, what we call it as an Easter cycle, which takes 4 to 5 days, but the menstrual cycle is 28 days, can easily comparable to the animals and the menstrual cycle. This is the experimental, and where you can see on the human being. And even you can see in the liver and other organs, these chemicals that will affect. This is the ones where we have done lot of work on experimental models, and these are the things where we discovered, and these are the standard methods we have used in the estimation of DNA genomic level, whether it is on the transcription or translation or at what level, or mRNA, where exactly these will act on this one, or whether it changes the nucleosides or nucleotides, where exactly it affects. That is the one. Among all the pesticides, as I already told you, that the chlorinated pesticides are much more dangerous. Even DDT can survive, or it can live without degradation more than 100 years. So by cloning process, now we have transplanted the genes responsible for degradation of these things at presently by using the genetic engineering. So where we have now identified new microorganisms, we transfer the genes, desirable character genes, and now we are trying our level best to degrade all those things. Now these are another the things, chemicals where we have discovered. So you can see the SDH, the LDH or 3 beta HSD or 17 beta HSD, and even the ATPases, acid phosphatase, alkaline. All these are the things where we analyzed, and we have also studied the oxidative stress parameters, and where SOD, catalase, peroxidases, so many enzymes we have studied how exactly these chemicals that acts when it enters into the body, and how it will have the even analysis of DNA in agarose gel also we have studied how it is affected. Now this is another very important part. This is the transfer section of the testis. So where you can see how the spermatogenesis that is there, you can see the seminiferous tubules, and lastly, last slide you can see so how these affected in the fifth slide at the below, and completely you will get the whole the seminiferous tubules without the sperms. So the sperms counts will be reduced, and this becomes the sterile animal. So this is how you see how much dangerous they are there. So when inhaled, it affects the hypothalamus. We will come later how the mechanism that exists in the animal. Now this is another agarose gel electrophoresis of cellular DNA in the testis of mite after the exposure to the methomyl, and you can easily see the mice treated with methopyl showed a sense of DNA indicates necrosis or the necrosis of the cell in the testis of mice. So you can see the bands, and that you can see how this is there, and that will be affected, the DNA that is affected. This is a very important mechanism that exists. See the hypothalamus that is present in the brain, and you will get the pituitary gland, that is the adenohypophysis, where you can distinguish into adenohypophysis, intermediate lobe, and the neurohypophysis, where these chemicals through the that is the releasing hormones or releasing proteins from the hypothalamus, it affects the adenohypophysis cells, and also the affecting that is the FSH, the LH, and that will act on the cells of the testis, where the testosterone production will be stopped, and naturally where the male characters diminishes and the sterility that causes. So that means it affects the hypothalamo-hypophyseal ovarian axis, and also it also affects hypothalamo-hypophyseal adrenal axis. So where these are affected, naturally the whole system is affected, and it also causes sterility. Now the hypothalamus, as just now I told, the GnRH, the gonadotropic releasing hormones, it acts on the pituitary, and the pituitary releases the LH and FSH, so where the testis is affected, and from the testis there is a production of testosterone. So in the feedback mechanism, this testis also produces a inhibin or a follistatin, where again it acts on the pituitary, it self-regulates the production of hormones. So this whole system, the feedback mechanism is affected, and naturally the production of testosterone is affected. So that is the one. Now this is the biosynthesis of testosterone. So in our section, one of the scientists were delivering his talk on coronary artery diseases, so where the lipids, or you can see the cholesterol, what it deposits, and it will affect the atherosclerosis. But I told that cholesterol is not at all, because some part may be there, because adipose tissue will be there in the blood vessels. This cholesterol is very much essential for the steroidogenesis. So otherwise the man becomes sterile. If the cholesterol completely, if you remove from the body, the whole steroidogenesis activity will go vanish. So that is how a part of it, maybe it will accumulate in the adipose tissue of the arteries or the coronary artery, it may leads to the atherosclerosis or whatever the coronary diseases that takes place. The doctor has fully agreed that yes, what you told is correct, and where the cholesterol is not only that is mainly responsible, but other things also responsible for the coronary artery diseases or atherosclerosis, where it leads to cardiac failure. Now this is the whole mechanism. You can see the testosterone, even 3 beta HSD, 17 beta HSD, all these enzymes are responsible for steroidogenesis, which takes place in the Leydig cells of the testis. So Leydig cells of the testis also will be affected, because these Leydig cells are mainly under the control of the LH. And also the seminiferous tubules also will be affected, and it is again under the control of follicle stimulating hormone produced from the adenohypophysis, and where in turn now the testis produces the testosterone, by this method you can see lastly the testosterone can be synthesized. So these chemicals affects the steroidogenesis and that leads to sterility. So that is how the dangerous chemicals, now what we see even in vegetables, even in water, even some of the soft drinks also contains these chemicals that is affecting the whole. So now compared to earlier reproductive potential I am telling, even in the male and the female, this reproduction potential is reducing day by day. So earlier even the sperm counts also decreasing day by day. Earlier the reproduction capacity is one entirely different, but presently now this is goes on decreasing because of so many things. Even excess of alcohol that also causes the sterility. There are so many factors that is responsible for sterility, but even drugs also. If you take the drugs, so without any proper advice of the doctors, these drugs are also affecting the whole system where it leads to sterility. Now this is another one. So this is how, what is the mechanism that exists? So okay, these chemicals when you take, so how the mechanism that affects, you can see the carbamates can interfere with the catecholamine neurotransmitter metabolism by inhibiting the activity of dopamine. This is very important. Beta hydroxylase, that is D-beta-H. This is an enzyme that converts dopamine to norepinephrine, and norepinephrine then stimulates the release of GnRH. Thus, the GnRH release is affected through the inhibition of delta beta hydroxylase. So where and so that will affect the whole system. This is what we have studied in the brain cells, that is the neurons, where we have analyzed this one, where it affecting this system and affecting ultimately the LH, which is mainly responsible for the production of testosterone. Now this is another very important, this is the mechanism just now I explained. This is another very important. So now any chemical, any metabolic or anything that enters in your body, it will end up with the Krebs cycle. So this Krebs cycle leads to electron transport system. So if this system is correct, perfect, then it will be converted into carbon dioxide and water. If it is not converted, then it will affect the whole system. Even whether it is action oxidative enzymes or let it be anything, so even the glycolysis or Krebs cycle, all electron transport system which takes place in the mitochondrial particles, that will be affected, and naturally the whole system collapses and there is no production of ATP. So just imagine, so how these chemicals are much more dangerous to the human being. Now this is another very important mechanism that you can see. So though these chemicals that lock on the LDH or SDH activities inter-converting, or you take the pyruvic acid, which is the end product of the glycolysis, and this pyruvic acid, if it is aerobic, then enters into the Krebs cycle where it converts into carbon dioxide and water. If it is anaerobic, then it may convert into any compounds in the body, maybe lactic acid, maybe any other anaerobic compounds that will be produced in the body. So that is how this whole system also that will be affected, and that is the one, it will enter into the citric acid cycle. Now the possible mechanism for the carbamate induced oxidative stress, and this is the possible mechanism, so where the carbamates you can see reactive oxygen species, where the lipids, proteins and the DNA molecule also affecting, and that leads to cell death, or you can say the mutagenicity, or it leads to carcinogenesis. So this carcinogenesis, when it induces the cell, the normal cell, when it transforms into a cancer cell, then you can see it is very difficult to control. First and second stage, usually we identify at the third stage, very difficult to control it. So the cancer again is the, every cell in our body have the capacity to transform into a cancer cell. But the only thing, where the cell divides fast, the attack of cancer will be more. And that is how, where the division is more, for example, you take the esophageal tract or bone marrow or uterine endometrium or cervix, you take where the cells divide fast, the attack of cancer will be more. So even the muscles, very less. Even in brain also, so where the brain tumor formation will be less is, but the cells where it divides, because the DNA exposed will be more, so the attack of the cancer or transforming the normal cell into a cancer cell will be more. Now this is the reactive oxygen species where they will be produced in the body to inhibit. So our body produces the catalase, or you say the that is SOD, that is superoxide dismutase or peroxidases, to control, to mobilize these chemicals into the body. So this is another very important one, where we also worked on the ovarian hormones that is produced in the female individual. So now a very important hormone is the estrogen. So the estrogen, which is produced from the granular cells of the ovary, that controls the whole mechanism of the woman. So where the menstrual cycle, the fertility, everything, so even implantation, pregnancy, lactation, parturition, everything is controlled under these hormones, the estrogen and the progesterone. The estrogen produced from the Graafian follicle or the granular cells of the Graafian follicle, and the progesterone that is produces from the corpus luteum of the ovary, where they produces these hormones that will mobilizes the fertility in the woman. So these are also affected, and that leads to again amenorrhea or oligomenorrhea, that leads to sterility. So these are the because all the system that exist in the body, the reproduction plays a very important role compared to the other system, because once the man or any animal becomes sterile, the continuity of life stops. So that is how this is very important one, where we have studied on the follicle cells, lot of work we have done on this, and that is the work. This is the mechanism that exists in the hypothalamus and the hypothalamo-hypophyseal gonadal axis, where that was the testis and this is the ovary. You can see the transverse section of the ovary, and that is the hypothalamus, and that is the pituitary gland, how the LH, RH, FSH, all those things that maintains ultimately the estrogens and the progesterone that controls the uterine endometrium, that controls the menstrual cycle. So these hormonal imbalance or the changes affects the. Now this is another one, hypothalamic pituitary ovarian axis. So how these are the things that takes place. Ultimately the last one is estradiol 17 beta, that is because you will get three hormones, estrone, estradiol 17 beta and estriol. Among all these three hormones in the female, estradiol 17 beta plays a very important role in maintaining the secondary sexual characters, and also the other, even fertility, everything is controlled by this hormone, which is a very, very important. That is how it is a female hormone. That also will be affected because these are the steroidogenesis or the steroid hormones. When these chemicals act, naturally the synthetic activity of this capacity, it reduces. So that also affected, and that is another mechanism that you can see. So even in other parts, we have also studied on the liver. Liver will do 1,000 and odd functions, how the liver is affected, and sometimes it leads to liver cirrhosis. So that will have so much of adverse effects on the body, and that is, you can see how these liver cells, the hepatic cells that will be affected by these chemicals in the last dose. And this is the kidney. Kidney also very important. You take in the diabetic patients or any other patients, because kidney is a very important organ in our body, where most of the toxicants that is produced or metabolized is removed by the kidney. So this is why in diabetic patient goes on removing the glucose from the blood, and how much stress it will you are giving to the kidney, and naturally ultimately the kidney will be affected. At the end the kidney will fail. So this is because of continuous functioning of the filtration of the all waste products. So from the glomerulus, where the filtrate takes place and where it removes all the waste products, even excess thing that is present in our body. That is how the glucomate, which the tablets, where the glucogenesis, because the diabetes is very dangerous disease, where the liver, the pancreas, these two organs plays a very important role in controlling the diabetes. So that is there. Now, now this is the agarose gel electrophoresis of cellular DNA in the liver of mice after exposure to methomyl. How you can see the stream of DNA that is affecting, and that leads to necrosis or it leads to the apoptosis. Now the agarose gel electrophoresis cellular DNA in the kidney that we have studied, and even the other biochemical, this is also very important one, where we have studied in the behavior of the animal. So we are keeping so many experiments we have done. So these chemicals we have given, so you see the behavioral changes, how it affects the brain cells. So that is how even what the tobacco also very dangerous, because gutka, all those things what the man takes, very dangerous, I will tell you, because not only it affects the neurological cells, and where it leads to so many diseases. This is the animal and different experimental, this Morris water maze with the SK platform, how when the person takes the alcohol, how it affects the cerebellum, the balance of the man is affected naturally by taking nicotine or any kind of the chemicals which enters into the body, even drugs also, that affects the brain cells. It is not advisable because even excess of anything, all those things, it is not advisable because it affects in the neuromechanism. So these are the elevated plus maze, all this. See, this is very important one. Very beautifully we got the results, and you can see, so in the hippocampus region we have taken the transverse section of the brain. And when you take the transverse section of the brain, you see the cells, how you can just see the epithelial cell, how it will be affected in the last slide. In the fifth slide, you can see how the control one in the top, and you can see how damages and where it affects the neurological disorders. This is another mechanism. This is the one in the high magnification, how this affects the hippocampus region of the brain cells. So all those things that affects and the one. Then later what we thought of, yes, if this is having so much adverse effects on the body, what happens in the nature? Then we started digging the things, we collecting the microorganisms from the soil, then we culturing these organisms and started our research work and what to do for this chemical. Then the biodegradation process we started analyzing. How to remove these chemicals from the nature when it has been sprayed, and what is the mechanism that exists. The nature take care everything. No doubt we add so many chemicals into the nature, but the nature take care of all these chemicals which you added into the nature. Just like mother, how she take care of everything, the son or the daughter do mistakes, exactly same thing. The nature is a mother where it takes everything and mobilizes the thing. Still then, the man interferes. Wherever the man interferes, you please remember, the whole thing collapses. So whenever you allow the man to interfere, so the system will collapse. So the nature, when the man enters and destroys the nature, ultimately we are facing a lot of problems because of man only. We have destroyed the forest, destroyed everything, ecosystem. So now these microorganisms, then we started analyzing how these microorganisms are affected, how we have studied in the higher organisms, the same thing what we studied in the lower organisms. That is the one which we discovered, Pseudomonas aeruginosa, that analysis from the nature, and that is also affecting the protein. This is the gel electrophoresis, what you can see this one. Then we started, can the genes responsible for this? Then we also started analyzing the genes that is responsible for this degradation process. Then what catalyze genes? There are 5 that is the genes will be there containing different elements for this one. We started digging, analyzing, and then we started the genes responsible for the one which degradation of this one by DNA sequencing. This is the one what we analyze, superoxide dismutase. This is a very important enzyme, not only present in the man, it is also present in the microorganism, that is Pseudomonas aeruginosa, where we analyzed from the nature and we got it. This is another one, catalase. So this is a sequence of the DNA. So where we have done, we analyzed this gene responsible for that is the degradation of methomyl. This is peroxidase. So these are the things where we started digging. Then we started remediation. What is it? Bioremediation, it takes place. So ultimately any chemicals, anything you added in the nature, it must convert into carbon dioxide and water. If it is not converted, if it is converted into any intermediate compound, this intermediate compound is much more dangerous than the original compound. So that is how we started analyzing. So how we have to bring it to the carbon dioxide and water. If it is not converting completely, then what we have to do? Then we have studied some cloning experiment. We transform the gene into this E. coli bacteria, which is a very important one, where we also transfer the desirable gene, that is the one which produces the insulin. So the insulin gene we have transferred and we are got it, that is expressing the human gene. So just imagine that even some of the, we are in plant biotechnology, some of the human genes has been transferred into the plant. We have transferred into potato or tomato. If insulin you want to produce, if the man unable to produce, gene therapy comes into picture. Till now, we are not on the gene therapy because so many drawbacks are there. We talk about if the insulin producing gene is mutated, if it is not producing insulin, then you transfer the gene into the beta cells of islets of Langerhans. But it is not possible because it affects the feedback mechanism. So once it start producing, more excess of insulin is start producing, controlling capacity will not be there. excess of insulin, it leads to insulin shock. Exactly, now we are unable to bring the gene therapy into consideration. So where we are unable to do lot of work on gene therapy, there are so many progressing work is going on the gene therapy. So now here also the gel electrophoresis what we analyzed, and now, so this is what we have started, how to cure this one. Now what happens in bacteria? So you will have the plasmid. So the plasmid will be there, the genome will be there. So both are involved in the degradation process. Means they produces the enzyme, and this enzyme cuts these chemicals and produces the carbon dioxide and water. So ultimately now, which is where exactly it acts, whether the plasmid is involved in the degradation process or the whole genome is involved in the degradation process. Then we started digging these things. Then what? How to remove the plasmid? Then we started removing the plasmid from the bacteria and allowing only the genome to work. So when allow to genome to work, and some of these experiments what we have done, in some of the pesticides, the plasmid is mainly responsible for the degradation process. And in some of the chemicals where the main genome is responsible for degradation process. But in some of the microorganisms what we see, both the genes are present in the plasmid as well as in the genome for the degradation process. So where both are involved in the degradation process of these chemicals. That what we studied under this, the plasmid curing experiments we have done on the Pseudomonas aeruginosa. Then studies on another chemicals we also studied on imidacloprid. So where we also analyzed the degradation of these chemicals. So bioremediation by the soil microorganisms. This is another very dangerous chemicals where they use farmer, and this is the ones where we have analyzed all those things, we cultured, and ultimately, so we also started analyzing of the protein again. So where this also very dangerous chemicals, then here what we observed is the plasmid is responsible for the degradation of this imidacloprid. So like that, so many chemicals we started analyzing, and also we studied the genome analysis. So which gene is responsible for the degradation process, which protein or which enzyme is responsible for the degradation process. That also we started. These are the different proteins they expresses, and that proteins are responsible for the toxicity, and these again we studied on this one. Other all enzymes also we have studied, what we have studied, and phylogenetic things we have analyzed, and also these are the some of the chemicals. So gel electrophoresis, we started analyzing the gel, the DNA and other chemicals, so where we also started studying. And another one again, the different chemical microorganisms we started analyzing from the nature, and how exactly they act, whether the plasmid or the main genome is responsible of the one. This is another one. So now this is the ones where one of my PhD students has worked on this, the propiconazole. Lot of work he has done for his, and his PhD viva is kept on 13th of this month. So where he is going to give a viva on this chemical, a lot of work, and he has published, and docking system in the bioinformatics. He also studied which site is responsible. So lot of research, isolation of DNA and which gene is responsible, chemical analysis, all those things, FTRI, all the experiments he has done. So these are the different methods he has followed, and effect of propiconazole fungicide on human cell line. So then he also studied whether these proteins or the enzymes which we discovered also helpful in the curing of cancer. So he has also studied on cell lines, and where the inhibits some of these things, and cytotoxicity test of these chemicals also we have studied, where it shows the cytotoxicity, and the determination of maximum inhibitory concentration value we also studied, and these are the degradation process that takes place, and this is quantification of pyrazole we have done in the LCMS. All the chemical analysis we have done. A bit by bit we have analyzed, and what is the toxicity of these intermediate compounds we have analyzed. Extraction of metabolites by preparation of HPLC. HP also we used, what is the micro concentration of these chemicals, and we analyzed all these metabolites, everything, and we also given the sequence how first time in the kinds where how this propiconazole that will degrade into the carbon dioxide and water. And gene identification also we have done, SOD, CAT, all these are the genes analysis what we have done. This is the one, and this is the docking system where we have studied in these microorganisms. One of the topmost work he has done on the bioinformatics, and this paper is accepted in US journal where the impact factor is 6, and where he has published the data, docking system, active sites, all those things that he has studied. And so where we are able to tell that this is the mainly responsible for the degradation process. With these few words, friends, and also the budding scientist, so my appeal is that so many things that is added in the nature, to protect the nature, that is very, very important, the message. You should not use as you wishes. Nobody is there to tell you that how to control these use of these chemicals into the nature. If you protect the nature will protect you. If you destroy the nature, you are destroying yourself. So with these few words, really I thank the organizers and the ISCA for giving an opportunity to present my work. This is just 2 or 3 PhD students' work. It's a lot of other works also there on the cancer and the diabetes and other things. So that because of time short, and I have presented a little, so 0.001% of my work, and where my students have taken a lot of pain in analyzing, in identifying the genes, genomic levels, and all those things. I thank for the award, and also I thank all of you for patient hearing.